Hello everyone, welcome to my channel Anupma Biology Classes. In this video we will see Kingdom MONERA part 1st, and this is the representative of the 5 kingdom system of classification, and this is the first kingdom. This video is taken from CBSE class 11th, chapter 2nd, and this is part 3rd of the biological classification. Now let's start the topic. So the Kingdom MONERA includes the most ancient, the smallest, the simplest, and the most plentiful prokaryotes. So, therefore, the Kingdom MONERA is also known as prokaryota. In Greek, pro means before and kary means nucleus. That means they have false nucleus. At place of nucleus, nucleoid present. The term prokaryote means before a nucleus. That means this organism lack a true nucleus, that is, the nuclear material of these organisms is not enclosed within the nuclear membrane, and some basic proteins are absent in the nucleoplasm. And the Kingdom MONERA includes bacteria, cyanobacteria, archaebacteria, and some smaller groups like Mycoplasma, Rickettsias, Actinomycetes, etc. However, the differences in structural and morphological characters among these groups are so little that it is very difficult to distinguish the groups, subgroups, genera, and species of these organisms. Now, let's see the first part, that means the first group of MONERA, which is bacteria. Bacteria are minute, unicellular, microscopic, simple, undifferentiated prokaryotic organisms which are usually identified by their specific activities and not by their general appearance. Bacteria were discovered by a Dutch naturalist, Antony van Leeuwenhoek, who invented the first microscope in 17th century and observed different specimens including scrapings from his own teeth. He observed bacteria on June 10, 1675, but he thought them to be animalcules, like little organisms or little animals, due to their small size and motility. However, these organisms were termed bacteria by Ehrenberg in 1838. Later, in about 1850, Louis Pasteur led the scientific foundation of bacteriology, that is, the branch of science that deals with the study of bacteria, by developing culture techniques and methods of sterilization. Now, let's see here the general characters of bacteria. So, what are the general characters of bacteria? See here, first we see about the occurrence, then size, then shape, and last is the flagellation in bacteria. So, first is occurrence. Bacteria live anywhere. They occur in water, in soil and air. Some types live as saprophytes while others are parasites on plants, animals and humans, causing diseases. Some of them can survive in ice and others can live in hot water. They can form spores which are very resistant to drought, chemical rays and temperature variations. So bacteria are ubiquitous. That is, they occur anywhere and everywhere, in water, in air, in soil, in all living organisms except the pits of volcanoes, deep strata of rocks, tops of high mountains and blood of healthy animals. Some thermophilic bacteria can withstand the temperature up to 78 degrees centigrade, while some psychrophilic bacteria occurs up to the temperature of minus 190 degrees centigrade. If we talk about the size of bacteria, the average size of bacterial cell ranges from 1 micrometer to 10 micrometer in length and from 0.7 micrometer to 1.5 micrometer in width. The size of smallest bacterium, an example, Koki, is not more than 0.1 micrometer, while the largest reaches up to 500 micrometer. So the largest bacterium is Bagiota mirabilis. It is 16 to 45 micron in diameter and several centimeters long. So the largest bacteria is Bagiota mirabilis and the smallest bacterium example is Cocci. Now in case of shape, the shapes and forms of individual bacteria usually remains constant. However, some of them are able to change their shape and size with changes in environmental conditions. Such bacteria which changes their shape are called pleomorphic. So the bacteria possess the different forms. Here you can see that is Cocci, Staphylococcus, Diplococcus, Bacillus, Vibrio, mycelial, stucked, budded, etc. So first one is the coccus or Cocci. Coccus is the singular and Cocci is the plural form. So the coccus bacteria are spherical, ellipsoidal or ovoid in shape. Here you can see all the structures of the Cocci. Coccus forms may exist singly, which is known as micrococcus, in pairs like Diplococcus, in tetrad like Tetracoccus, in chains like Streptococcus or in seeds like Staphylococcus. A few cocci may occur in cubical packets of 8 or more cells, which is known as sarcinae. Here you can see this is the structure of sarcinae. Now arrangement of bacillus. The bacillus bacteria, plural is bacilli, are straight, rod-shaped or cylindrical. The ends walk may be pointed, flat, concave or convex. They may occur singly, which is known as bacillus. Here you can see. In two, that is known as Diplococcus. Here you can see, this is the Diplococcus. Then like a stack, which is known as palisade bacillus. These are palisade. And or in chains like the Streptobacillus. After this, see here the spirillum. Spirillum, spira means coil. So the spirillum bacteria are spiral or coil like a corkscrew. The spiralar forms are usually rigid and bear 2 or more flagella at one or both the ends. Example like spirillum, spirochetes, etc. After this, see here that is the vibrio. The cursor represents this is the vibrio. The bacteria are rod-shaped with single curve, C-shape or comma shape. They have one or more flagella at one pole. Example is vibrio. After this, filament. The body of bacterium is filamentous like a fungal mycelia. The filaments are very small, like examples of filaments are Vagiota, Thiothrix, etc. Next is stalked. The body of bacterium poses like a stalk like the Caculobacter. And the last one is budded. The body of bacterium is swollen at places like rhodomicrobium. So these are the shapes of bacteria and these are coccus, bacillus, spirillum, vibrio, filament, stalked and budded. Now after this, see here flagellation. Before the flagellation, see here the shape of bacteria and the examples of bacteria. So first you know that the coccus and the examples are Micrococcus, Diplococcus, Tetracoccus, Streptococcus, Staphylococcus, etc. After this bacillus examples are Bacillus, Diplobacillus, Palisade Bacillus, Streptobacillus, etc. Vibrio example is Vibrio. Filament examples are Vagiota, Thiothrix, etc. Next is stalked and the example is Caculobacter and the last one is budded and the example is Rhodomicrobium. Now, see here the flagellation. So on the basis of number and mode of attachment of flagella, the bacterial forms may be divided into the following types and the first one is known as atrichous. Here you can see this is the atrichous, that means no flagella. That means flagella absent. An example is Lactobacillus. Next is monotrichous. That means one flagellum at one end. And the example is Vibrio cholerae. After this, see here, which is known as cephalotrichous. So what is cephalotrichous? See here, means many flagella attached to one end is known as cephalotrichus. An example is Pseudomonas fluorescens. Next is amphitrichus. See here. And the example is Nitrosomonas. Here the single flagellum attached to each of the two ends. So one flagellum at one end and the other flagellum at the other end. After this, Lophotrichus. See here. A group of flagella attached at each of the two ends. So which is known as Lophotrichus, like the Spirillum volutens. And the last is Peritrichus. So what is Peritrichus? Flagella are attached all over the body like Salmonella, typhi, E. coli, etc. So on the basis of type of flagellar arrangement, first one is the polar or monotrichus, that means single flagellum at one end. Next is Lophotrichus, that is stuffed of flagella at one end or one pole. Amphitrichus, that means flagella at both poles. Peritrichus, that means flagella all over the body. Amphilotrichus means tuft of flagella at both the end. And if the flagella is absent in bacteria, is known as atrichus. So this is all about the flagellar positions and the flagella on the body of bacteria. These are the locomotory organs. After this, bacterial cell structure. So a typical bacterial cell shows the following structural features, in which the first one is the slime and the capsule, which is known as glycocalyx, the cell wall, plasma membrane. In plasma membrane, mesosomes, flagella, pili or fimbriae, then cytoplasm and cytoplasmic inclusions, nucleate, it means nuclear body or bacterial chromosomes, and plasmids. So these are the structural parts of the bacteria. Now we will see each and every part in detail. So the first one, that is the slime and the capsule, which is known as glycocalyx. So slime is the gelatinous substance secreted by the protoplast and deposited over the cell wall in the form of a loose gelatinous sheath, which is called slime layer. It is usually composed of polysaccharides. In case of some virulent bacteria, the nitrogenous substances are also present in addition to polysaccharides, so that the slime layer becomes thick, called capsule. The capsule is usually, here you can see, this is the structure, that means known as glycocalyx outer layer, which is known as capsule. So the capsule is usually found in parasitic forms like the Bacillus, Diplococcus, Mycobacterium, where it provides the protection against phagocytosis and antibodies. Now see here, that is the slime layer. The slime layer or capsule also protects the cell against desiccation and viral attack. The capsule bacteria are usually non-flagellated. It means atrichous, like Lactobacillus. Now see here, the cell wall. The bacterial cell wall is the outer rigid, like this is the cell wall of bacteria. This is after the capsule. So the outer layer is capsule, after this cell wall, and after this plasma membrane, and inside the plasma membrane cytoplasm is present. So the bacterial cell wall is the outer rigid and tough covering of the cell, which provides shape and protection to the protoplast and keeps the cell from bursting or collapsing due to osmotic changes. It ranges in thickness from 50 to 100 angstrom. Chemically, it consists of acetyl glycosamine, acetyl muramic acid, and a peptide chain of 4 or 5 amino acids. All these chemicals together form a polymer called peptidoglycan or murine or mucopeptide. Some other chemical substances deposited on the cell wall are teichoic acid, protein polysaccharides, lipoproteins, and lipopolysaccharides. Now see here the gram-positive and gram-negative bacteria. So the cell wall of gram-positive bacteria is much thicker and contains less lipids as compared to that of gram-negative bacteria. The enzyme lysozyme can dissolve the bacterial cell wall. After this, the plasma membrane. Each bacterial cell has plasma membrane situated just internal to the cell wall. It is a thin, elastic and differentially or selectively permeable membrane that allows passage of dissolved substances in and out of the cell. It is composed of large amounts of phospholipids, proteins and some amounts of polysaccharides but lacks sterols. The plasma membrane of bacteria provides sites for the most of the anabolic and catabolic metabolic pathways. It is characterized by possessing respiratory enzymes which are bound to its inner surface. Some exoenzymes are also associated with its outer surface, which catalyze digestion of insoluble materials. Now, the structures associated with the plasma membrane, in which the first one is the mesosome. See here, this mesosome structure attached with the plasma membrane. This is the plasma membrane. The outer green color is the cell wall, and after this the outer will be glycocalyx. So see here, what is the mesosome? In many bacteria, especially gram-positive, the plasma membrane gives rise to infoldings called mesosomes. These are finger-like, spherical or villiform structures which may help compartmentalize the cell. Mesosomes participate in the formation of septa during the cell division and secretion of intracellular substances from inside to outside. They also take part in the separation of replicated nucleoid. After this, see here the flagella. This is the flagella. You can see the large snack-like structure which is red in color is the bacterium flagellum. So, these are long thread-like structure concerned with the motility of some bacterial cells. Each flagellum measures about 4 to 5 micrometer in length and 12 to 15 micrometer in diameter. A flagellum consists of identical 3 to 8 subunits called flagellin, arranged helically along the axis to give a hollow tube. It has no definite membrane. The flagellum is attached to the cell membrane by a special terminal hook, which is turned in attached to the basal body. The 360-degree rotation of flagellum includes the cell to spin and move forward. After this, see here the pili or fimbriae. So this is the pili, which is also known as fimbriae. These are extremely minute, non-flagellar but straight hair-like appendages. They measure about 0.5 to 2 micrometer in length and 3 to 5 nanometer in diameter. These are composed of protein subunits called pili arranged helically around a central hollow core. The function of pili. This is the pili. The function of pili is not in motility, but they help in attachment of the bacterial cells. Some sex pili act as conjugation canals through which DNA of one cell passes into the other. Pili have been observed mostly in gram-negative bacteria. Now, after this, cytoplasm and cytoplasmic inclusions. So, the cytoplasm. What is cytoplasm? The cytoplasm is a complex aqueous fluid or semi-fluid ground substance which is known as matrix consisting of carbohydrates, soluble proteins, enzymes, coenzymes, vitamins, lipids, mineral salts and nucleic acids. The organic matter is in the colloidal state. The cytoplasm is granular due to presence of a large number of ribosomes which occur singly or in small groups which is known as polyribosomes. The ribosomes in polyribosomes are held together by means of messenger RNA. The ribosomes of bacteria are smaller as compared to those of eukaryotic cells. Membranous organelles such as mitochondria, endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles are absent. In some photosynthetic bacteria, the plasma membrane gives rise to large vesicular thylakoids which are rich in bacteriochlorophylls and proteins. Now, nucleoid. What is nucleoid? Also known as nuclear body or bacterial chromosome. The true nucleus with a nuclear membrane is absent in bacterial cell. The nuclear material consisting of the naked DNA molecule is identified as nucleoid or bacterial chromosome. It is a circular, ring-like, double-strand DNA molecule with no free ends and measure about 1000 microns in length. Unlike eukaryotes, there are no histones, that means basic proteins, in the nuclear material. But recent findings indicate the presence of at least two proteins which bind DNA. The circular DNA is attached at a point to the plasma membrane. During replication of the bacterial nucleate, the mitotic apparatus is not seen. Now, after this, see here the plasmid. What is plasmid? In addition to bacterial chromosome, many bacteria like E. coli have accessory rings of DNA which is known as plasmid. The plasmid DNA replicates independently, maintains independent identity, and may carry some important genes like fertility factor or F factor, resistant factor or R factor, nitrogen fixing genes like NIF genes, etc. The plasmids which temporarily get associated with nucleoid DNA are known as episomes. So this is all about the structure of the bacterial cell. Now, after this, see here the bacterial reproduction. How bacteria reproduce. So, bacteria can reproduce by two means. First one is asexual reproduction by binary fission, and the second is the sexual reproduction, which is genetic recombination or parasexuality. And they have three parts, that is conjugation, transformation and transduction. Now let's see each and every part in detail. So in case of asexual reproduction, bacteria reproduce asexually by means of binary fission. Other asexual methods of reproduction are budding, conidia formation and sporulation, etc. So first see here, in case of binary fission, it is the most common method of asexual reproduction in actively growing bacteria. The cytoplasm and nuclei divide equally into two without mitosis. During the division of nuclei, the DNA replicates while attached to mesosome. A new mesosome develops which gets attached to the daughter chromosomes. So, at the same time, the cell wall and the plasma membrane start growing transversely inwards from near the middle of the division cell between the two mesosomes. So, the dividing septum originates centripetally and separates the parent cell into the two nearly equal daughter cells, each having a nuclear body. So, these cells may separate from each other or may remain attached for some time to form chain. So, here you can see there are some positions like the parent cell, then replication of DNA, then segregation of DNA, and the last one is cell splitting into two. So binary fission is a process where one cell divides to form two identical cells. This is an example of asexual reproduction. Under favorable condition, the process of binary fission completes in about 20 to 30 minutes. Thus, a very large number of bacterial cells may be produced in 24 hours. So in case of binary fission, steps are: first one is the cell elongation and DNA is replicated. Then cell wall and plasma membrane begins to divide. After this, the cross wall forms completely around divided DNA and then cell separates to form offsprings. So this is all about the asexual reproduction, and this is the binary fission of bacterial reproduction. Now after this, see here the sexual reproduction in case of bacteria. So sexual reproduction in bacteria is not of the kind of found in eukaryotic organism. In case of bacteria, the sex organs are not formed. Meiosis and mitosis does not occur. The two gametes do not fuse with each other and the diploid zygote is not formed. What is diploid zygote? Having two sets of chromosomes within a true nucleus. So instead, a portion of genetic material, that is DNA, is transferred from a donor cell, that means male, to a recipient cell, that is the female, making it an incompletely diploid zygote. The process is actually called genetic recombination, which occurs in three ways. First one is conjugation, then transformation, and then transduction. So first one, that is the conjugation. Transfer of DNA by the process of conjugation occurs between two sexually different strains of bacteria. One acts as a donor of the genes, that is male, and the second is the recipient cell. That means that means female. So both are haploid. During the act of conjugation, the genetic material is transferred from donor to the recipient through a conjugation bridge formed between the two. So in this case, see here, then the transfer of a fraction of a gene, DNA, that means genes, extracted or otherwise liberated from a donor bacterium to a recipient bacterium is called transformation. During this process, a very small amount of DNA is transferred intracellularly from one bacterial cell to the another bacterial cell, where it gets integrated with the DNA of recipient bacterium. The recipient bacterium cell then acquire the characters encoded by the foreign DNA and pass it to the offspring. So the addition of a foreign deoxyribonucleic acid DNA into a bacterium is known as transformation, and transformation was discovered in 1928 by Frederick Griffith. After this, the transduction. Transduction is the process in which the genetic material, a portion of DNA of one bacterium, is transferred to another through the agency of temperate lysogenic bacteria. The process was discovered by Ginder and Lederberg in 1952 in bacteria Salmonella typhimurium. So in this case, first is the phase injects DNA, then the phase enzyme breaks down host DNA, then cell creates new phages including phase and host DNA, then transducing phase inserts donor DNA, then donor DNA includes in recipient chromosomes due to recombination. So this is the sexual reproduction, that means genetic recombination or parasexually. We will see each and every part. So in the case of complete process of sexual reproduction, which means genetic recombination, first process is conjugation, which is a direct transfer of genome. Here, the genetic material of a bacterial cell of a specific strain is shifted from a donor or male into that of another bacterial cell, recipient or female of a different strain. The donor cells have a sex factor or F factor, and the recipient cell do not possess this factor, and thereby it is expressed as F strain. Now the second is transformation. Here the DNA from one bacteria is transferred to the another bacteria. And the last one, that is the transduction. Here the DNA is transferred from one bacterial cells to a different bacterial cell through a bacteriophage virus. It is known to take place in different bacterial species, namely E. coli, Micrococcus, Salmonella, etc. So this is all about the sexual reproduction in case of bacteria. Now, after this, see here the economic importance of bacteria. So, in case of economic importance, there are some useful activities and some are harmful also. So, useful activities are decay of organic waste, sewage disposal, role in nitrogen cycle, role in improving soil fertility, role in industry, medical uses, etc. And the harmful activities are food poisoning, spoilage of food, pollution of water, deterioration of textiles, and diseases. So, now let's see each and every activities in detail. First is the useful activities, and in which the first one is decay of organic wastes. Many saprotrophic bacteria act as natural scavengers by continuously removing the harmful organic waste from man's environment. They decompose the organic matter by putrefaction and decay. The simple compound produced as a result of decomposition and decay, like carbon dioxide, carbon monoxide, nitrate, sulfate, phosphate, ammonia, etc., are either released back into the environment for recycling or absorbed by the plants as food. Thus, the bacteria play a dual role by disposing of the dead bodies and waste of organism and by increasing the fertility of soil. Now, sewage disposal. Ability of anaerobic bacteria to putrefy the organic matter is used in the sewage disposal system of cities. The feces are stored in covered reservoirs and allowed to putrefy. The solid matter is decomposed into the liquid sludge, which is passed through coarse filters. The effluent is finally putrefied and drained out into the river or used as fertilizers in the field. The common bacteria involved in sewage disposals are coliforms, streptococci, clostridium, micrococcus, proteus, pseudomonas, lactobacillus, etc. Now, role in nitrogen cycle. Nitrogen cycle existing in nature comprises of conversion of atmospheric elemental nitrogen into usable forms. Second is ammonification, then nitrification, then denitrification. Bacteria play a very important role as they are involved in almost all the steps of nitrogen cycle. Role in improving soil fertility. Saprophyte bacteria present in soil perform various activities for their survival. Some of these activities improve the fertility of soil by formation of humus, manure, etc. So, first one, that is the humus. The microbial decomposition of organic matter and mineralization results in the formation of complex amorphous substance called humus. The humus improves the aeration, water holding capacity, solubility of soil minerals, oxidation reduction potential and buffering capacity of the soil. Now, in case of soil fertility, composting. It is the conversion of farm refuse, dung and other organic waste into the manure by the activity of saprophytic bacteria. And nitrogen fixation, many free-living and symbiotic bacteria and nitrogen content into the soil by the process of nitrogen fixation. Now, adding sulfates. A few sulfur bacteria add sulfur into the soil by converting H2S into the sulfate. So, these are the role in improving soil fertility. Now, after this, see here the role in industry. Useful activities of various bacteria are employed in the production of number of industrial products like first one is the lactic acid. Lactic acid is commercially produced from pasteurized whey, the watery part of milk, through fermentation caused by Lactobacillus bulgarius and Lactobacillus delbrueckii. In case of curd, curd is prepared from pasteurized milk by the process called curdling. It is initiated by adding a starter culture of Lactobacillus bulgaricus and Streptococcus thermophilus into the milk at 40 degrees centigrade. Lactobacillus converts lactose to lactic acid, whereas Streptococcus causes coagulation of casein due to acidity. In case of cheese preparation from the milk involves too many steps. First, curdling of milk, and second, the subsequent ripening of solid curd by the use of different bacterial strains. In butter is prepared by churning of sweet or sour cream. The microorganisms responsible for preparation of buttercream are Streptococcus lacti and Leuconostoc. The bacteria, the characteristic butter aroma develops due to a volatile substance diacetyl. Then vinegar. Country-made vinegar is the fermentation product of cane juice, molasses or fruit juices. It is produced in two steps. First, conversion of sugar into alcohol by alcoholic fermentation carried by yeast, and the second, conversion of alcohol to acetic acid by the action of bacteria Acetobacter. Rating process. What is rating process? Fibers of flax, hemp and jute are separated by the process called rating. During this process, the stems of the plants are submerged in water, where the bacterial activity resulting in the rotting of softer parts. The tough bast fibers becomes loosened and easily separated from each other. These fibers are spun and woven into various articles. Then, curing of tobacco and tea. The curing of tobacco and tea leaves is carried by fermenting action of certain bacteria to add special flavor and taste. Coffee and cocoa are also prepared by the action of certain bacteria to add characteristic color, flavor and aroma in them. After this, leather tanning. The process of tanning hides and skins after drying, salting and clearing involves the fermenting action of certain bacteria obtained from excreta of poultry, dogs, etc. In case of medicinal uses, first one is the serum and vaccines. So many bacteria are used in the preparation of serums and vaccine, and these substances induce immunity to various diseases in men. Serums are effective against certain diseases like diphtheria, pneumonia, etc., whereas the vaccines are effective against typhoid, smallpox, cholera, etc. Now, in case of vitamins, production of riboflavin, vitamin B2 involves the activity of bacterium Clostridium botulism. The well-known vitamin C, ascorbic acid, is produced from sorbitol by the action of Acetobacter species. Some bacteria live in the alimentary canal of herbivorous animals like cow, horse, goats, etc. and help in production of certain enzymes which digest the cellulose. In case of antibiotics, these are several chemical substances produced by the living organism capable of inhibiting or destroying other microbes. These are the products of secondary and minor metabolic pathways, mostly secreted extracellularly by the microorganism. They are used in controlling various infectious diseases. So these are the useful activities of the bacteria. Now after this, see here the harmful activities. First one is the food poisoning. Some saprophytic bacteria causes decay of our foods, that is, they alter their normal form and induce unpleasant aroma, taste and appearance. Some bacteria produce powerful toxin in food to cause food poisoning. Like botulism. Botulism is caused by Clostridium botulinum. The main symptoms are vomiting followed by paralysis and death. After this is staphylococcal food poisoning, which is caused by Staphylococcus aureus. Common symptoms are nausea, vomiting and diarrhea. Then see here perfringens poisoning, which is caused by Clostridium perfringens. Symptoms appear in the form of diarrhea and acute abdominal pain. Now, spoilage of food. Some examples of bacterial food spoilage are greening on meat surface is caused by lactobacillus and leuconostoc. Salmonellosis is poultry and eggs is caused by salmonella. Black rot of eggs is caused by proteus. Green rot of eggs is caused by pseudomonas. Red rot of eggs is caused by Serratia. Rofi bread, having soft and cheesy texture with long string threads, is caused by Bacillus. Souring of milk is caused by Lactobacillus and Streptococcus. A explosion of curd, that means gas production, is caused by Clostridium and coliform bacteria, and Ropinase, that is slimy milk, is caused by Klebsiella species, Enterobacter species. So these are the spoilers of food. Now, pollution of water. There are reports of epidemic of cholera, typhoid, jaundice and other infectious diseases which were caused by polluted water. Many pathogenic bacteria such as Vibrio cholerae, Salmonella typhi, Shigella dysentery, Leptospira, etc. are pollute water and make it unfit for drinking. These are eliminated by chlorination. Now, deterioration of textiles. Some bacteria like Vibrio cellulomonas damage cellulose of textile. And then some diseases which are caused by bacteria are the causative. So bacteria are the causative agents of a large number of human diseases such as pneumonia, typhoid, dysentery, cholera, plague, influenza, tetanus, diphtheria, tuberculosis, leprosy, syphilis, whooping cough, etc. They are also responsible for several plant and animal diseases. So this is all about the first, that is the bacteria. What is bacteria? And their role, their activities, reproduction, characters, etc. Now in the next video we will see actinomycetes and cyanobacteria also. So this is the end of bacteria. So if you understand this video, please like and share it and subscribe to my channel and my biology classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.